Hi everyone, my name is Taeyu and I'm a junior at St. Paul American School, Hanoi. I co-lead the H2O Science Club at our school, and today I'll be walking you through the chlorophyll chromatography experiment. This is a classic AP biology lab that connects directly to Unit 3 of Cellular Energetics. We'll explore the science behind pigments in photosynthesis, how chromatography works, and finally, how you can conduct this experiment step by step. So before we get into the experiment, let's talk about the background science. Photosynthesis is the process plants use to convert light energy from the sun into chemical energy stored in glucose. This happens inside chloroplasts, and this requires pigments to capture light energy. Without these pigments, plants wouldn't be able to absorb sunlight effectively. Plants don't just rely on one pigment. They have multiple pigments that each absorb light at different wavelengths. Chlorophyll A is the primary pigment and looks blue-green. Chlorophyll B is an accessory pigment that's yellow-green. Carotenoids give orange colors and xanthophylls are yellow. All of these pigments work together to maximize the plant's ability to capture solar energy. Each pigment has a unique absorption spectrum, meaning it absorbs certain light wavelengths of light better than others. For example, chlorophyll absorbs blue and red light but reflects green light. That's why most plants look green to us. Having multiple pigments allows plants to capture a wider range of the light spectrum, boosting their efficiency in photosynthesis. Now you might wonder, if leaves look mostly green, where are the other pigments? They're actually hidden under the dominant green color of chlorophyll. Chromatography allows us to separate these pigments so we can see the full variety inside the leaf. This gives us insight into how plants are biochemically adapted to capture light energy. Chromatography is a laboratory technique used to separate mixtures of molecules. It works because different molecules dissolve differently in the solvent and stick differently to the paper. Pigments that are more soluble in the solvent will travel faster, while those that stick more strongly to the paper will stay closer to the origin line. That's how we can separate and identify the pigments. Now let's move on to the experiment. The main purpose of this experiment is threefold. First, to separate the pigments found in spinach leaves like chlorophyll A and chlorophyll B, carotenoids, and xanthophylls. Second, to understand these pigments absorb different wavelengths of light, allowing plants to maximize their ability to capture solar energy. And third, to practice chromatography, which is an essential lab technique that is used for separating molecules. Here are the materials you'll need. Most of these are common lab supplies: chromatography paper, mortar and pestle, toothpicks, scissors, and isopropyl alcohol as the solvent. Safety goggles are essential since isopropyl alcohol is volatile and flammable. Before we start, let's talk safety. Always wear goggles because we're working with alcohol. Handle the solvent carefully, keep it away from flames, and make sure containers are closed when not in use. Lab safety always comes first. Moving on to the procedure. First, we cut up a strip of chromatography paper about 2 centimeters wide. Draw a light pencil line around 2 centimeters from the bottom. This will give us our origin line where the pigment will be placed. Now crush spinach leaves using a mortar and pestle until you have a fine paste. Dip a toothpick into the paste and draw a thin line of pigment on the origin line of your chromatography strip. Repeat this about 5 times to make the band darker and concentrated. Next, pour about 15 milliliters of isopropyl alcohol into the graduated cylinder. Attach the chromatography paper strip to a pencil with tape and hang it so the bottom just touches the alcohol. But make sure the pigment line does not get submerged. Cover the top with plastic wrap to prevent evaporation. Now let the chromatography run. Over time, the alcohol will move up the paper through capillary action, carrying the pigments with it. This process usually takes hours or even overnight. Once the solvent is near the top, remove the strip and quickly mark the solvent front with a pencil. After drying, you'll see a distinct pigment bands. Carotenoids are yellow to orange, xanthophylls are yellow, chlorophyll A is blue-green, and chlorophyll B is yellow-green. These bands represent how far each pigment traveled based on solubility and attraction to the paper. To make this quantitative, we will calculate RF values. Measure the distance from the origin line to the center of each pigment band and divide it by the distance from the origin line to the solvent front. This number is always between 0 and 1, and it will help us to identify pigments more precisely. The science behind this is really cool. Different pigments dissolve differently in the solvent and stick to the paper with different strengths, which is why they separate. Accessory pigments like carotenoids and xanthophylls expand the range of light a plant can use, making photosynthesis more efficient. In conclusion, this experiment gives us a hands-on way to understand how plants are adapted for photosynthesis while also practicing important lab skills and scientific reasoning. Thanks for watching.